In this talk, we are going to learn about axonal guidance.
Now let's look at axonal guidance. Now the axons have to find their correct locations, and the way they're able to find their correct locations usually involves three steps. The first one is pathway selection. So axons travel along a route that leads them to a particular region of the embryo, basically where their targets are. This process does not require neuronal activity, which basically means that we don't need the neuron or the axon to transmit action potentials. The second step is target selection. So, on reaching the correct area, the axons will recognize and bind to a set of cells with which they may form stable connections. And this process also does not require neuronal activity. The final step is address selection. So in this step, the initial patterns are refined such that each axon will bind to a small subset, like even only one of its possible targets. Now this process requires neuronal activity. Now let's look at the first step, which is pathway selection. In this case, we see the movement of the axon towards its target. Now we should remember that neurons in similar places have similar targets. And in many cases, the location where the axon must go is specified before the axons are formed. So for example, in the case of motor neurons, the motor neurons know where they're supposed to go or where their targets are before the axon is formed from the cell body. This was demonstrated by an experiment where we have a developing embryo and certain parts of the embryo, especially in the posterior part where the motor neurons will come, those parts were actually switched. So we have this T7 to LS3, and what we can see is the portion of T7, LS1, 2, and 3 were switched. And that is what is shown in the figure that is there below. So in this case, this is normally how the motor neurons would form, and they form from the T7 and LS1 region, and we can see the way they are crossing over. Now when the whole pattern was reversed, what we notice is even though the pattern was reversed, the motor neurons went to the same position and not to another position. That way, these developing motor neurons knew where they were supposed to go even before their axons were formed. Now, the Hox and limb protein families aid in target specification, and the initial direction of growth is mediated by integrins and encadherins. Adhesive substrates like laminin in the extracellular matrix also aids in movement of the axon towards its target. And finally, existing axons also help in movement. Let us look at how existing axons help in movement. Now, the axons of existing neurons can direct the growth of other neurons. This has been shown in the case of sensory neurons. Now, the sensory neuron axons actually rely on the motor neuron axons for guidance. So, the process where one axon adheres to and uses another axon for growth is called fasciculation. And this is what we observe in the case of sensory neurons. So, sensory neurons use motor neuron axons and hence they fasciculate. And this is what is shown in the figure in the yellow box, where the green is the motor neurons and the red are the sensory neurons. And the sensory neurons basically are moving using the motor neuron axons as guidance, and this is what we can actually see in this figure where we see the yellows. Now, the process of movement is guided by molecular street signs, which are basically molecules. And we're going to look at four main molecules that play a role in axon guidance. They are the ephrins, the semaphorins, netrins, and slit proteins. Now, here we have a figure where in the blue box, we can actually see how some of these molecules help in the process of axonal guidance. In most cases, ephrins and semaphorins repel the axons, and hence they're able to guide them by preventing the axons from going to certain places. Here is an example where motor neurons have been stained, And what the box is showing the presence of motor neurons, which are these dark orange structures that are present in the anterior portion of the sclerotome. These are present in somites, and we had seen the sclerotome when we had studied about the neural crest migration. Now, when we look at the caudal or the posterior portion of the sclerotome, we don't see the presence of the motor neuron axons. This is mainly because in this region we have the efferents and semaphorins that prevent the axons from coming to the posterior part. Another example where we can see is where the neurons are developing from the limb bud and are going to the central nervous system. In this case, we can see the portion in the blue box is where we have cells that are making semaphorin. And due to the presence of semaphorin, we can see that the axon has actually changed its direction and is moving downwards to cross that particular region that has the semaphorin secreting cells. Thus, efferents and semaphorins tend to repel axons, and hence the axon structures try to move away from those regions. Now, we should remember that in some cases, efferents and semaphorins can act as attractants for other structures, for example, dendrites. If we look at the figure that's there towards the right, we can see a neuron, which is an NT3 responsive neuron, and right next to the neuron, to the right of it, is a semaphorin secreting cell. And we can see that the dendrites that are emanating out of the cell bodies are basically not bothered by the presence of the semaphorin 3. So in this case, we do not see a repulsive effect of the semaphorin. However, when we look at another type of neuron, like the mechanosensitive neurons, in this case, we can see that the dendrites don't want to be close to the cell that is secreting the semaphorin 3. Thus, depending on the type of neuron, we can see different effects of these semaphorins and also efferents. But however, for the purpose of this class, it is enough to remember that both efferents and semaphorins are repulsive in nature. Another set of molecules that aid in axonal guidance are sonic hedgehog and netrin. Both of these are secreted factors and they act as attractants, so axons like to go towards sonic hedgehog and netrin. And when we look at the figure that is there to the right, we can see that we're looking at two neurons and they're extending their axons, and their axons are going towards the bottom part. And that is where we see more of sonic hedgehog and netrin. And that is why the axons are actually attracted towards that posterior region or that lower region, and hence they are moving in that direction. Another protein that is secreted is called a slit. This protein mediates axon guidance and acts as a repellent. It prevents the axons from crossing the midline of the nerve cord. Now, this is what is shown in this figure where we can see the expression of slit. Wherever we see the dark brown line, that is where the slit protein is present. And the slit binds to another receptor called roundabout. So neurons tend to express the receptor roundabout, and roundabout will bind to slit. When we look at the expression of roundabout, which is also abbreviated as ROBO or ROBO, we can look at its expression, and when we compare the expression of the slit and the roboprotein in a developing embryo, we can see that they are not next to each other, but they are in different locations. Let us look at how slit and robo play a role. Now, many neurons have to cross over from the right side of the body to the left side of the body and vice versa. Slit and robo aid in this type of crossing over to ensure that it is done appropriately. So let us look at how this happens. Now, neurons, the axons of neurons, they tend to move towards the midline through the actions of netrin and sonic hedgehog. And as we saw previously, netrin and sonic hedgehog are attractants. And so in the midline, we see more of netrin and sonic hedgehog, and hence the neurons tend to move. And that is what we're seeing here, where the axon is moving towards the midline. Now, when the axon is near the midline, it downregulates the expression of robo. The reason it does that is because in that midline region, that is where we have the expression of slit. So the cells in that midline are expressing slit. And we had seen that central expression in the previous slide when we looked at the expression of slit in a developing embryo. So as the axon comes to the midline, it actually will downregulate the expression of robo. Since it does not have robo expressed, it will not sense the presence of slit and hence it can move and cross the midline. So the important point to remember in all of this is even if you have a paracrine factor or any kind of a factor, that factor cannot mediate its effects on cells until there is a receptor on the cells to sense it. So in this case, robo is the receptor that senses slit. If there is no robo, the cell cannot sense slit. And hence that is what is happening with the axon, where the axon that is now going towards and crossing the midline has downregulated the expression of robo, and hence it is unable to recognize slit, and slit has no effect, and thus the axon crosses over. Now, after crossing over, let's see what happens. Once crossing over has happened, robo is expressed in that growth cone. And now that robo is expressed in that growth cone, this axon cannot come back, and hence it remains on the other side of the midline. Thus, when we look at the picture that is shown towards the right, when we look at a wild type animal and we look at the way the axons of the neurons are stained, we can see this ladder-like arrangement. However, if you do not have slit present, then that is what is shown in panel D. In the absence of slit, we don't see proper crossing over occur because there is no way to tell the axon that it does not have to come back and that it has to stay where it is. Thus, slit and robo play an important role in these crossing overs of neurons from the right to the left. Now let us look at what happens to the axons once they are at their final destination. So at the final destination, the axons are able to find their appropriate target cells by attractive and repulsive signals. Now the target cells are usually muscle cells or are another neuron. Now the growth cones are able to select their targets based on the combined input of attractive and repulsive signals. And hence it is not just one signal that plays a role, but a combination that plays a role. The examples of some molecules that play a role in aiding the growth cones to their target cells are endothelins. So these are small peptides secreted by blood vessels. And thus, if we have neurons that need to come near the blood vessels, the endothelins play a role in guiding their axons to the appropriate target cells. Another example are neurotrophins. These are molecules secreted by target cells that could be attractive or repulsive depending on the kind of neuron and the kind of target cell. So thus, this way, the axon is able to find its final destination through the chemotactic proteins that are made by the target cells. Once the axon is able to find its target cell, a synapse has to be formed. So a synapse is a specialized junction that is formed between the axon of the neuron and its target cell. The steps involved in synapse formation are, the first step is where the axon contacts the cell membrane of the target cell. The axon terminal then accumulates neurotransmitter containing vesicles. Then the membranes of the target and the axon, they start thickening. The synaptic cleft, which is where the axon and the target cell are meeting, fills with an extracellular matrix. And these steps are shown in the figure that is there below. In the next step, the muscle-derived laminin, which is in the extracellular matrix that has been laid down in the synaptic cleft, it binds to the growth cones to signal the axon to stop growing. Now, the growth cones from other axons are now able to converge to form additional synapses. Now, this is what is shown in the figure that is there in the blue box. So once one axon makes contact with the target cell, we can have axons from other neurons, so different neurons also converging to that area, and that way additional synapses are formed. Now, the axon that is able to display most neural activity will persist. It will also expand and get myelinated by the Schwann cells if this is a part of the peripheral nervous system. And that is what is shown here in this box. So, in the previous step, we saw that we can have axons from different neurons come to the same target cell. However, there will be a testing mechanism that happens to determine which axon is most efficient at transmitting the electrical impulses. And only that axon will persist. That way, in the end, a target cell will have only an axon from one neuron. And so the other neuronal axons will go ahead and they will die out. Thus, in this step, we need neural activity to determine which axon will persist. And that axon that persists will then expand to form more branches. It also gets myelinated by the Schwann cells so that now you have an effective synapse that has been built. Now, during normal course of development, over 50% of the neurons will die in many parts of the central and peripheral nervous system. The type of death that occurs in these cells are programmed cell death. So a way to look at this, or another way to say this, is that during the normal course of development, we make way more neurons than what we actually need. So we're making more than 50% of what we actually want. Thus, we need to get rid of all the extra neurons, and for that, cell death is necessary. Apoptosis, which is the programmed cell death, is necessary for the proper spacing and the orientation of neurons. So in the previous slide, we had looked at how, in spite of axons from different neurons coming and interacting with target cells, how only one of them persists. The other that does not persist will basically die from apoptosis. The proteins involved in apoptosis include FF1 and many caspase family members. Apoptosis has been covered in depth in cell biology, so please go through that process in case you need to revise how apoptosis actually occurs. Now, when we look at why apoptosis is necessary, let us look at the figures. So in the first figure, we're looking at a wild-type embryo, and it shows all the normal features and focus on its head region. Now, if we take another developing embryo where one of the apoptosis proteins is knocked out, which is caspase 9, in this specific embryo, we don't have apoptosis happening very well. And what we can see is if we focus on its brain region, we can see a very enlarged brain. And that is because the neurons that were supposed to die have not died, and hence you have excess neurons present, which is not a good thing because you don't have good electric impulses being transmitted properly. These animals will basically die and they won't even be born. Thus, it is important to go ahead and have most of the neurons that do not function properly to die by programmed cell death. Now, neuronal survival is dependent on neuronal activity. So the successful synapse formation is required for neuronal survival. If we don't have a successful synapse formation and an electrical impulse that is being generated, the neuron will not survive. Target cells promote survival of neurons through the action of neurotrophins. So once a successful synapse is formed, the target cell can aid in the survival of the neurons by secreting or making the neurotrophins. This is what is seen in this figure. So if we look at neurons in the sympathetic ganglion, we can actually see the effect of two neurotrophins. The The first one is NGF, which actually promotes the survival of the neuron. However, when we look at another type of neurotrophin, which is BDNF, that one does not actually allow the survival of the sympathetic ganglion. In the case of the dorsal root ganglion, we can see that NGF and BDNF don't have much of an effect. They both promote the survival. However, when we look at the taste or the neurons that allow us to perceive taste, NGF does not promote survival, but BDNF does promote survival. Thus, the target cells have to make the appropriate neurotrophins to aid in the survival of the neurons.
To summarize, we learned about the different steps involved in axonal guidance, which includes pathway selection, target selection, and address selection. We also learned about mechanisms involved in the survival of neurons.